A person might question what organisms are living inside of their navel. While a person may consider this to be disgusting, it is part of our normal microbiota. Normal microbiota are microscopic organisms that normally reside in or on our body and are generally beneficial in maintaining homeostasis. Normal microbiota keeps organisms in balance and therefore help maintain health. If they become out of balance, then the organisms can become pathogenic. The purpose of this lab is to isolate and identify some of the bacteria living inside of the navel. It is necessary to first isolate each individual bacteria in order to run the proper testing needed to identify them. Identification is done by analyzing the various tests performed that separate the bacteria according to the differences and similarities of structure, chemical composition, and metabolic capabilities. These tests include the use of differential stains, differential and selective media, and metabolic tests. In order to detect differences in structure and chemical composition of organisms, a differential stain is used. Differential media detects what different bacteria metabolize. Selective media will tell which organisms favor or inhibit the media. A study done by a group of North Carolina researchers found 2,368 species of bacteria present, with 1,458 species of these new to the scientific world. Their test included complex DNA analysis. In the present study, our group was able to isolate and identify 6 different bacteria, including Staphylococcus aureus, Streptococcus phalanges, endospores, Salmonella, E. coli, and Serratia marcinescens.
I'm going to tell you about our test results. When it comes to the gram-negative organism test results, we had 3 unknowns, and all 3 of them were bacilli. So at that point, we knew that we would have to do more testing, so we decided to do the EMB growth plate. Unknown 1 came back as green, and unknown 2 and 3 came back pink. That presents a problem because with them both being bacilli and pink, we knew that we would have to do another test. So we decided to do the MRVP test on them, and 1 came back positive, 1 came back negative. And since those results were found, we went to Berkey's manual and found out that we had Serratia marcinescens and Salmonella. We wanted to be sure that we had Salmonella, so we decided to do the heptoin enteric, and we came back with the blue test result, which was positive for Salmonella. Once we got the green EMB growth on unknown 1, we decided to do a catalase test, which came back positive, and we at that point deducted that we had E. coli. But since we were already doing the MRVP test, we decided to test it for E. coli, and it came back positive. And the heptoin enteric also came back yellow, positive for E. coli. So because of those test results, that is what we determined that we had for the gram-negative organisms in the belly button. For the gram-positive, we had, again, 3 unknowns. 2 of them were cocci and 1 was bacillus. We knew at that point that we would have to do more testing, and we knew that from the colony morphology, we had a yellow, white chains, and green. All 3 different, but still we had to do more testing. So we did an MSA plate, and it came back positive on unknown number 4. We still needed more information, so we did an OF test that came back positive and a catalase test, and it came back to be Staphylococcus aureus. For 5 and 6, there was no MSA testing to be done or OF test. But for unknown number 5, we got to do a catalase test, and it came back negative. And because of those testings, we determined it was Streptococcus pyogenes. For unknown 6, they were endospores. So once you determine that it's bacillus and the colony morphology is green from the acid fast test, that's all you can do. And again, with our results, that's the conclusion that we came to. Of course, we could have used DNA if that was available to us at Carl Sandburg, but unfortunately it's not. So this is the results that we came up to with our gram-positive organisms, and we hope you've enjoyed our little presentation. Thank you.